Hi, I'm Kajal.
Hi, I'm Sana.
As for tracing associated receptor 1, there's a total of 6 in our bodies. It helps cure brain disorders, and it's found in the stomach, kidney, lungs, and blood. It plays a role in neurotransmission, so it impacts serotonin, dopamine, and norepinephrine levels. It also plays a role in Parkinson's disease. An example would be muscle spasms that are caused. Non-epinephrine would be similar to adrenaline, which is an excitatory response, and it relates to arousal and memory. And another example would be dopamine. It contributes to sufficient rates of metabolic processes and digestion. It regulates the amount of neurotransmission is present. It's dependent upon cognition and its impacts. It increases CAMP, which is used for signal transductions, and it binds well with the TAAR1 receptor. The family is traceamines. They're basically like G-group proteins. They detect molecules outside of the cell. It activates responses and transductions. It passes through the plasma membrane exactly 7 times, and there's limited amounts available. Pharmacology of traceamines, it relates to serotonin, which is mood, and adrenergic, which is basically adrenaline, so epinephrine and non-epinephrine. And it secretes neurons that connect with dopamine, non-epinephrine, and serotonin, and it's prevalent in brain disorders.
Now we're going to talk about our 2 main neurotransmitters. So neurotransmitters are chemical messengers, and they link the brain and spinal cord with muscles, organs, and glands, which we call the receptor sites. It regulates a wide variety of processes such as fear, pleasure, anger, a lot of your emotions. This is a diagram that shows from one neuron to the next neuron, because neurons don't actually touch. They have a gap in the middle, which is called the synaptic cleft. So how neurons communicate is through these chemical messengers, which are neurotransmitters. So the types of neurotransmitters. You can have excitatory and inhibitory. So excitatory means that an action potential is likely to happen, and an action potential is basically the probability of one neuron firing to the next neuron and then the next neuron, which lets the message continue to send. Inhibitory is when an action potential will probably not occur. So there are 15 G protein-coupled receptors that we have in our bodies, and an orphan receptor PNR, which is just another type of a receptor. When they're combined together, you get a subfamily of rhodospin GPCRs, and it belongs to the biogenic amine receptors, which is basically a fancy word of saying neurotransmitters. So 3 of these receptors are present in the amygdala, and the amygdala is the center of our brain that controls fear and other emotions. So this is evidence that these receptors do play a role in diseases that control emotion. So a type of neurotransmitter receptor is a ligand-activated ion channel, and this produces a very quick physiological response, because from the moment a neurotransmitter binds, as you can see, from the minute a neurotransmitter binds, ions will be released, so current will start to flow. The minute the neurotransmitter detaches, the current will stop and ions will stop flowing, therefore depolarization will occur. So some serotonin receptors are ligand-activated channels. Another type of neurotransmitter receptor is a metabotropic receptor. So the signaling is much slower because there's an additional step. They use second messengers. So as you can see, there will be a neurotransmitter that does connect, but there's also 2 additional pieces. This is just sort of floating, but then that's your second messenger, such as maybe CAMP, and that's going to be used in producing a response. And both dopamine and serotonin use metabotropic receptors. So neurotransmitter structure. So they interact with G protein-coupled receptors, as we've talked about, and both are biogenic amines. They contain a carboxyl group, which is like amino acids, but they lack a carboxyl group, which is unlike amino acids. And dopamine is synthesized from the amino acid tyrosine, and serotonin is synthesized from the amino acid tryptophan. So dopamine. Dopamine is both inhibitory and excitatory depending on which receptor it goes to. It's important in reward and pleasure. It's important for memory and motor skills. And balance is very critical when it comes to dopamine levels, because if you have too much dopamine, you get a dopamine storm, and that results in hallucinations, which can lead to schizophrenia. If you have too less dopamine, it results in muscle tremors, and you have no control over your muscles. So that's why people with Parkinson's usually have muscle spasms and they are not able to control their tremors. Balance is very critical. And this diagram is just an example of a human dopamine that is found in our bodies, and it's actually from the PDB. So serotonin, on the other hand, is mostly inhibitory, and 95% of it is found in the digestive system. And it's actually, that's why from our gut and below is called our second brain, because it controls a lot of different functions. Your brain and your second brain are highly connected because of serotonin. It's important in mood and digestion. So we talk about serotonin with mood a lot, because if you have low levels of serotonin, it can lead to depression. This is another diagram that we got from the PDB, and this is human insulin in complex with serotonin. Like I said, serotonin is found in the digestive system a lot, and that's why it is in conjunction with this insulin.
Now we're going to connect everything together. So what we learned about TAAR1, neurotransmitters, and diseases. So phentermine is just an example of a trace amine. It's not necessarily the only one that can trigger this process, but basically phentermine is dissolved in the blood, and then ligands that make up phentermine are going to bind to the TAAR1. So this right here is just a very basic diagram of what happens. Your R is going to be your TAAR1 receptor. This G is your G-coupled protein, and the E is the enzyme that the TAAR1 is going to help activate, and we'll talk more in depth as I go through this. But basically, the phentermine is going to bind to the receptor site on the TAAR1. So this would be, this hormone is technically phentermine. It's going to bind to TAAR1, and it becomes a GPCR. So that's when it becomes a coupled protein, because another protein is added to it. So these two together is a GCPR, and that kind of triggers an active state. And the GCPR is made up of alpha, beta, and gamma subunits, and that alpha subunit originally has a GDP molecule attached to it, but once it binds with TAAR1, that GDP is replaced with GTP, which activates only the alpha subunit. So then the alpha subunit, which is now active, is going to bind to this enzyme, and it's going to activate that enzyme using the GTP enzyme.
Alpha subunit of the GCPR is activated, and therefore activates this enzyme called adenosine cyclase. And adenosine cyclase is basically going to stimulate the production of CAMP. And CAMP is a precursor to ATP, but in this case we're using ATP to make CAMP. And CAMP's going to bind to these receptors, these little orange bubbles, and they're called PKA, which is protein kinase A. And protein kinase A triggers monoaminergic transporters. And monoaminergic transporters are simply that, they transport monoamines. And monoamines are essentially neurotransmitters, and we discussed that earlier. So basically, PKA stimulates the release of dopamine as well as the reuptake of neurotransmitters like dopamine. Now let's talk about the impact on diseases of the TAAR1 protein. The TAAR1 gene binds to trace amines, and now we're going to explain what trace amines exactly are. Trace amines are highlighted in blue in this diagram, and the structures highlighted in green are monoamine neurotransmitters. And here we can see that their structures are very similar. Dopamine and serotonin do have that extra OH group, and that OH, that hydroxyl group, is necessary for processes like dehydration synthesis and hydrolysis, which encourage the building up and breaking down of molecules, which suggests that while the trace amines, which are found at low concentrations in the brain, can't make those molecules, dopamine and serotonin can, which is key to their structure and their function. Provides evidence for the fact that TAAR1 is related to trace amines and is related to neurotransmitters, which helps validate the correlation that different levels of neurotransmitters and the regulation of the TAAR1 gene will influence neuropsychiatric disorders.
Impact on diseases. So basically, as I said before, TAAR1 helps activate these monoamine transporters, right? So it allows for the reuptake of dopamine. So once these transporters allow like dopamine to enter the synaptic cleft and be received by this neuron will take some of the dopamine, but some of it will not be taken up by the second neuron. So it has to be retaken up by the first neuron, which fired in the first place, in order to regulate your dopamine levels and make sure that you don't have a buildup of dopamine.
This is
found in cocaine addiction. This picture actually demonstrates that perfectly. The molecule cocaine in the brain will block these transporters, resulting in excess dopamine in the synapse. And as a result, patients feel a high, and that is what they're addicted to. In conclusion, the TAAR1 gene has significant impact on many different neurological disorders, such as schizophrenia, depression, Parkinson's, and addiction. And it also could possibly play a role in breast cancer. Recent research suggests that the TAAR1 gene can also help regulate potent immune functions, and these immune functions are extremely relevant to breast cancer cells. And if we could somehow control those through the TAAR1 gene, we could maybe even find a cure for cancer.
So, how exactly does this all relate to chemistry, though?
So basically, the TAAR1 receptor protein in the membrane of the neuron needs to bind to dissolve phentermine, and phentermine is a trace amine. When it dissolves, the bonds break, and it's kind of in an aqueous solution, which is what we learned in class, how aqueous solutions, ions dissociate and then form new bonds and everything. So once that phentermine is dissolved, it can form new bonds with the trace amine associated receptor 1, right? And then that will alter the structure of the protein, and we've discussed the structures. So
We have. So, you're right. It does relate to chemistry.
yeah.
Thanks for watching!